Hey everyone, welcome back to this channel, and today we'll be discussing about the TCA cycle. As we all know, we have covered the glycolysis, pyruvate oxidation, and also gluconeogenesis and fermentation, and today we'll discuss about the TCA cycle. So first, Sir Hans Adolf Krebs. He was the scientist who first discovered this TCA cycle, or tricarboxylic acid cycle, and based on his name, this TCA cycle is also known as Krebs cycle. Okay, so till now we know that whenever we take glucose or carbohydrate through our diet, it enters into our blood. Okay, so blood glucose level is there, and also muscle glycogen, that means glucose gets stored into glycogen, right? And these two are the major source for glucose in cell. And then we have already shown that the glucose will undergo glycolysis, and from glycolysis two major things will be produced. First is the ATP, and second one is the pyruvate. Okay, and from this glycolysis, these two are the major final product of glycolysis. And then from this glycolysis there will be two parts. One is when there is no oxygen, so we can say that is the anaerobic condition over here. And if that is the anaerobic condition, the fermentation will be the fate. So fermentation will go on, so alcohol fermentation or lactic acid fermentation will go on. The second fate is if oxygen is present, and then the aerobic respiration will go on. And this TCA cycle is basically the phase of aerobic respiration. Okay, so where oxygen is needed or aerobic condition is needed. Don't get confused over here. TCA cycle doesn't require oxygen, but it should happen when oxygen is present. Okay, because after TCA cycle we'll see about oxidative phosphorylation, where the electrons have they have to be transported to oxygen so that water molecules can be produced. And in that phase of this carbohydrate metabolism, ATP is synthesized through ATP synthesis. That is the major source of ATP produced in our cell, and that obviously happens in mitochondria. So this is the aerobic respiration. This is the phase where TCA cycle happens. TCA cycle doesn't require direct oxygen, but it always occurs in presence of oxygen. Okay, so in aerobic condition this TCA cycle will occur, but TCA cycle doesn't require any oxygen, so remember that. The next thing is, obviously I have already mentioned that from this TCA cycle we gain ATP, GTP, NADH and FADH2. ATP and GTP, they are like the powerhouse of the cell. ATP mainly, this is the source of power for metabolic energy, cellular energy and everything. And NADH and FADH2, they act as the reducing agents. Okay, so they are the electron sources. Whenever we'll study electron transport system, we'll see that this NADH and FADH2, they are the major electron sources, so they are the reducing agent. And what about the location? I have already mentioned that TCA cycle happens in the mitochondria. Okay, so in the next phase we'll see the tricarboxylic acid cycle. As I have mentioned over here that there are 8 enzymatic reactions. Okay, so let's start with this part here. Acetyl-CoA, if you can remember, the acetyl-CoA was produced from pyruvate oxidation. Okay, so from pyruvate oxidation through pyruvate dehydrogenase complex, this acetyl-CoA is produced. So this is the first step of the tricarboxylic acid cycle. And here what is happening is this acetyl-CoA is releasing this CoA group, okay, as CoA SH, and with the help of citrate synthesis and H2O, this oxaloacetate and acetyl-CoA, they are like combining to each other, and it is forming the citrate, right? And then in the next phase this citrate is releasing another water molecule, and it is converting into an intermediate called cis-aconitate. The enzyme responsible for this reaction is aconitase. Okay, and since this is within the third bracket, means this is the intermediate, this is not a stable form, so cis-aconitate will be converted into isocitrate. See, Whenever citrate get converted into cis aconitate, there is a dehydration reaction, so H2O gets out. But whenever cis aconitate is converted into isocitrate, one H2O molecule is getting in. Then why this is citrate and this is isocitrate? See, there should be some difference in some structural difference, right? See, this is the first carbon, second carbon and third carbon. Let me see if I can zoom in a bit. Yeah. See, this is the citrate, this is the first carbon, this is the second carbon, and this is the third carbon, right? So the first carbon contain 2H2, okay, 2 molecules of hydrogen atom. The second carbon contain OH, and the third carbon contain H2, okay. But if we see in this isocitrate, see the difference. The difference is first carbon is same. First carbon containing H2. The first carbon here also the first carbon contain H2. The second carbon contain OH in citrate, but in isocitrate the second carbon contain H only. Okay, CO minus, these three are common in both citrate and isocitrate. Just focus on the left part in this part. Okay, just focus on the left part of this C. So first carbon is, it contain H2, this also contain H2, this contain OH, this second carbon of isocitrate contain H, and the third carbon of citrate contain again H2, but this isocitrate, the third carbon contain H and OH. So this is the difference between citrate and isocitrate. Just the H and OH is different in second and third carbon. Okay. Now when this phase is done, the isocitrate will get converted into alpha ketoglutarate, and the enzyme which is associated with here is isocitrate dehydrogenase. Since there is a dehydrogenase, obviously there will be an oxidation reduction reaction, and also there is one carbon dioxide molecule will be released, and so this is a decarboxylation reaction. So the CO2 molecule gets released, but from which carbon? From this second carbon, this CO2 molecule get released. If you see the alpha ketoglutarate structure, you will see that the CO minus is not present in the second carbon. So CO2 molecule is getting released from this second carbon. And the enzyme which is associated is isocitrate dehydrogenase. Okay, and NAD plus is converted into NADH and H plus. Okay, so after that this alpha ketoglutarate will be converted into succinyl CoA, and this is again another decarboxylation reaction. So CO2 molecule is getting released, and alpha ketoglutarate dehydrogenase is the enzyme. Dehydrogenase means again there will be some oxidation reduction reaction, and over here NAD plus is again converted into NADH and H plus. So these two successive steps, like isocitrate to alpha ketoglutarate and alpha ketoglutarate to succinyl CoA, this contain some similarities, like NAD plus is converted into NADH and H plus in both these cases. In both these cases the enzyme is dehydrogenase. For the first step, like for conversion of isocitrate to alpha ketoglutarate, the enzyme is isocitrate dehydrogenase, and the conversion of alpha ketoglutarate to succinyl CoA, for this conversion the enzyme is alpha ketoglutarate dehydrogenase. Okay, and the NAD plus is converting into NADH plus H plus, and in both these cases or both these steps CO2 molecule is getting released. So this is decarboxylation and decarboxylation plus oxidation reduction reaction, both is happening. Okay, and ultimately from alpha ketoglutarate succinyl CoA will be released. Okay, so in this step, like when isocitrate was converting into alpha ketoglutarate, the CO2 molecule was released from the second carbon of isocitrate, right? Now from the alpha ketoglutarate and succinyl CoA structure, we can see that this CO2 molecule has been released from the carbon 3 or third carbon of alpha ketoglutarate. This carbon has been released. Okay, so this carbon has been released. Here we have CO minus, here we have SCOA. Why SCOA? Because another molecule of CoASH is coming. CoASH means coenzyme A is coming. That's why this is the name is succinyl CoA. So this CO2 molecule gets out, and in this place, instead of this CO2 molecule, now an SCOA or CoA SH molecule has been added. So the name is succinyl CoA. Okay. So the next step is the conversion of succinyl CoA to succinate. Okay, succinyl CoA to succinate, and the enzyme which is associated with this reaction is succinyl CoA synthetase. Remember, this is not succinyl CoA synthase, this is synthetase. And one molecule of GTP will be converted into one molecule of GTP. So now this GTP can easily get converted into ATP, so there is no issue in that. Okay, so that's why sometimes we say that TCA cycle produce ATP, or we also may say that TCA cycle produce GTP, although GTP is more like accurate answer, so we should stick to that GTP molecules are being synthesized. GTP, NADH and FADH2, these three molecules are synthesized from TCA cycle. Okay, so by succinyl CoA synthetase, succinate is produced from which compound? From succinyl CoA. So succinyl CoA is being converted into succinate by succinyl CoA synthetase, and in that reaction GDP is converted into GTP. In the next step, we can see that again there is a dehydrogenase enzyme, and so there is again another oxidation reduction reaction, and in this case succinate is converting into fumarate, and one molecule of FAD is converting into one molecule of FADH2. Okay, again this FADH2, just like NADH, it will be acting as a reducing agent and will also be acting as like source of electron in case of the next phase, that is oxidative phosphorylation. Okay, let's stick to TCA cycle for now. So succinate is converted into fumarate by succinate dehydrogenase enzyme, and one molecule of FAD is converting into one molecule of FADH2. In the next phase, fumarate is converted into malate by an enzyme called fumarase. Okay, for fumarase we can see that we need H2O molecule, so one molecule of water molecule is needed for this reaction. So fumarate is being converted into L-malate by the enzyme fumarase, and finally this malate is converted into oxaloacetate by malate dehydrogenase. Again another oxidation reduction reaction, because dehydrogenase is the enzyme, and over here also NAD plus is converted into NADH and H plus, so that is malate dehydrogenase converting malate to oxaloacetate. Now this oxaloacetate will again be converted into citrate with reaction to a acetyl CoA. So acetyl CoA and oxaloacetate will react, and in presence of citrate synthase it will produce citrate. So that is a circular reaction that goes on and on and on, and total 8 enzymatic reactions are needed. Total 8 enzymes means first is citrate synthase, second is aconitase, as you can see, because this is an intermediate, so both this 2A and 2B we can say that these are being catalyzed by aconitase. The third enzyme is isocitrate dehydrogenase, very important enzyme. The fourth one is alpha ketoglutarate dehydrogenase. Fifth one is succinyl CoA synthetase. Sixth one is succinate dehydrogenase. Seventh is fumarate, and eighth is malate dehydrogenase. So these are the total 8 enzymes which are needed, and this is the total TCA cycle. Now we often face an answer that which carbon is released as CO2, right? So which carbon is releasing as CO2? As we can see over here that, see this was oxaloacetate, so we can see that the third carbon, that is CH2CO minus, and the second carbon, that is C double bond O CO minus, this has been converted into COH CO minus. So in this citrate we have first carbon, second carbon and third carbon. This second and third carbon, it is being derived from what? Oxaloacetate, and the first carbon CH2CO minus, that is being derived from acetyl CoA, right? And in this step, when carbon dioxide is being released, we now know that the first carbon dioxide is being released from the second carbon of isocitrate, and the second carbon dioxide is being released from the third carbon of isocitrate. So what is the source of second and third carbon? The source of second and third carbon is oxaloacetate, not the acetyl-CoA. Acetyl-CoA carbon is the first carbon of citrate. Okay, so here this is the C1, this is C2 and C3, and it goes on like this is C1, this is C2, this is C3, this is C1, C2 and C3. So we know that, just remember this thing, that whenever in the first cycle of the TCA cycle, what happens is the first CO2 is being released from the second carbon of isocitrate, and the second CO2 is being released from the third carbon of alpha ketoglutarate, and both the second and third carbon is being derived from oxaloacetate. So that is the thing. Another thing is, we often face another question, that if we just put a radioactive marker at the carbon, so where will get the radioactive carbon ultimately? So if we put the radioactive carbon in oxaloacetate, let's say in C2 or C3, we all can see, we all now know that this C2 and C3 will ultimately get released as CO2. So if we have a radioactive carbon in this C2 of oxaloacetate or C3 of oxaloacetate, it will be released as CO2 in the first cycle of this Krebs cycle or TCA cycle. Okay, first cycle means this cycle is going on and on and on. So first cycle means we are assuming that the first acetyl-CoA and first oxaloacetate is coming, and they are reacting to form citrate. So that is the first cycle of TCA cycle. The second stage is when this oxaloacetate and acetyl-CoA is producing the citrate, and the citrate is again converting into L-malate, and malate is converting into oxaloacetate. So that is the second cycle. So in the first cycle, what will happen, that if we just mark this oxaloacetate C1 and C2 of oxaloacetate by radioactive marking, they will get released, but this acetyl-CoA carbon will not get released. Okay, so what will happen if we mark this C of acetyl-CoA, this carbon of acetyl-CoA? See, we can track this acetyl-CoA carbon till citrate, in cis-aconitate, in isocitrate, in alpha ketoglutarate, and in succinyl-CoA. But whenever it is converted into succinate, you see the structure is CH2CO minus, CH2CO minus. Can we now differentiate between these two carbon? We cannot differentiate between these two carbon, and we'll say that if we mark this acetyl-CoA with some radioactivity, then at this stage of succinate, it will not be separated. It will be like partially, it will distribute between these two carbon, and will not be able to identify which carbon is from this acetyl-CoA. Okay, so that is another thing that you should remember. The next thing is about the regulation. So this TCA cycle is tightly regulated to maintain the metabolic homeostasis, obviously, because there are too many things going on. 2 CO2 molecules are releasing, 2 NADH molecules, 3 NADH molecules are being produced, 1 FADH2 is being produced, 1 GTP molecule is being produced, so it must be tightly controlled. But what are the enzymes? I have already said that there are 8 enzymes, but among them there are 3 enzymes which are really important in terms of regulation view. Okay, so the first thing is citrate synthase. This is the first enzyme that regulates the citric acid cycle, and this act as a pacemaking enzyme. Okay, so this act as a pacemaking enzyme, and it is living in all living organisms. Obviously it is not living, I mean to say that in all living organisms this citrate synthase enzyme is present, and this is encoded by the nuclear DNA except in the mitochondria. So we all know that mitochondria, they contain their own DNA, and mostly this citrate synthase is being produced by the nuclear DNA, but in case of mitochondria, they have their own like mitochondrial genes for producing citrate synthase. Okay, the next enzyme is isocitrate dehydrogenase. Isocitrate dehydrogenase, which reaction is being catalyzed by isocitrate dehydrogenase? That is conversion of isocitrate into alpha ketoglutarate, and we now know that one molecule of CO2 is released from that step. Okay, so this is also known as digestive enzyme, and it is used to catalyze the oxidative decarboxylation of isocitrate into alpha ketoglutarate, and this is encoded by the mitochondrial protein, both mitochondrial protein and cytosolic protein. Okay, so both cytosolic and mitochondrial protein. Cytosolic protein means the proteins which are present in the cytosol, that means not in the nucleus. Okay, that is present in the cytosol. So that is isocitrate dehydrogenase, and the next enzyme is alpha ketoglutarate dehydrogenase. This is like the major importance of alpha ketoglutarate is in determining the metabolic flux through the citric acid cycle, because we all know that through alpha ketoglutarate dehydrogenase, the alpha ketoglutarate will be converted into what? Succinyl CoA, right? Succinyl CoA. So there will be CO2. One CO2 will be released, and one NAD plus will be converted into NADH and H plus. So that by that means it can control, or it can determine the metabolic flux. Okay, also it catalyzes the alpha ketoglutarate to succinyl coenzyme A. We all know these things, and this is allosterically regulated and catalyze the irreversible steps of TCA cycle, which are main point of this regulation. Obviously the allosteric control mean it is being controlled by some other factors. It is not only controlled by the substrate concentration or the product concentration, it is controlled by some other products. And what are the products? We'll see in the next slide. So yeah, now we have talked about the regulation, and we have talked about the allosteric regulation. And so what are the allosteric regulator? We'll now see. So there are some inhibitors of TCA cycle. See, TCA cycle will happen in the cell whenever the cell energy level is low. That means ATP level is low and AMP level is high. That time only the TCA cycle will happen. But what if we have enough amount of energy? That means we don't need, the cell doesn't need the TCA cycle if the ATP concentration is quite high. Okay, so obviously there are some feedback inhibition. Feedback inhibition means if ATP and NADH, FADH2, these things are, we can also say that GTP, NADH and FADH2. So these are the main products of TCA cycle. Okay, and if these products are present in huge amount in a cell, then the cell doesn't need to do the TCA cycle. So that is the feedback inhibition. That means the product, if the product is present in huge amount, it will inhibit the enzymatic reaction. So high level of ATP and NADH can inhibit the activity of isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase, limiting the flow of intermediates through the cycle. Clear? Why isocitrate dehydrogenase and alpha ketoglutarate dehydrogenase? Because obviously ATP and NADH or GTP and NADH, these are the products that is being catalyzed by isocitrate dehydrogenase and alpha ketoglutarate. Okay, we can also say that succinate synthetase where the GTP is being produced, that is also inhibited by ATP. Okay, the next thing is oxygen deprivation. So this is one very important thing, that is feedback inhibition. Okay, so that is one thing, feedback inhibition. The next thing is oxygen deprivation. We have already discussed that this TCA cycle, it doesn't require oxygen directly, it doesn't require oxygen, but the medium, the environment should be aerobic, otherwise the TCA cycle will not happen. Okay, so even if the TCA cycle happens, the oxidative phosphorylation will not happen. That means the final aim of TCA cycle is what? To do the oxidative phosphorylation, to run the electron transport system so that oxygen can be converted into H2O and ultimately ATP is produced. So if we have conditions like hypoxia or anoxia, that means oxygen is deprived, there is no fate of TCA cycle, so the TCA cycle will be inhibited. The next thing is nutrient deficiencies. See, if we have deficiency in vitamin B1, that is also known as thiamine, it can impair the activity of TCA cycle enzymes like alpha ketoglutarate dehydrogenase, because this thiamine or vitamin B1 act as a cofactor for alpha ketoglutarate dehydrogenase. Also, it has been found in some studies that thiamine is very important for the synthesis of alpha ketoglutarate dehydrogenase. So if thiamine or vitamin B1 is absent, that means both the function the structural stability and functional integrity of alpha ketoglutarate dehydrogenase will be inhibited. Next, we have some toxins and drugs. These are also some examples of toxins and drugs. We can see that fluoroacetate is the toxin which can inhibit this aconitase. Okay, arsenite is the substance which can inhibit alpha ketoglutarate, and malonate is the substance which can inhibit the succinate dehydrogenase. Okay, so these three are very important. Questions often come from these examples. Okay, and the next part is obviously which is beyond of our control, that is genetic mutation. So if there is any mutation in the proteins which are associated with the production of TCA cycle enzymes, that will obviously lead to TCA cycle inhibition. Okay, and it may be inherited, it may be acquired through somatic changes, but whatever it is, this is beyond our control. Right, most of them are beyond our control, except this nutrient deficiency. We can have a good amount of nutrient, we can inhibit the intake of these toxins and drugs, we can be in a place where subtle amount of oxygen is present, but this feedback inhibition, with healthy lifestyle, we can also control to some extent. We can also control this feedback inhibition, but this genetic mutation, this is beyond of our capacity. So we should be we can't do this if any genetic mutation happened to one of the enzymes, one of the proteins which are associated with the TCA cycle enzymes. Okay, so that is the thing, that is overall view of the TCA cycle. Hope that makes sense. And okay, so this is a pathway that I have made. This is very crude pathway. So as we can see that if we take this chocolates, that means glucose, that will first undergo glycolysis, and then it will undergo pyruvate oxidation, and from pyruvate oxidation, actually in this case there will be like one is present of oxygen and another one is where no oxygen is present. So if no oxygen is present, it will undergo fermentation, and there will be lactate fermentation or ethanol fermentation. But if pyruvate oxidation happens, that will lead to acetyl-CoA, and that acetyl-CoA will enter into this TCA cycle. This TCA cycle we have covered, but we also have another phase. Like every other successive steps has been covered. The gluconeogenesis has already been covered in the channel, and fermentation, we have also done fermentation. The next phase which is remaining is this oxidative phosphorylation, and this is the ATP, this is the powerhouse. Okay, the ATP formation, that is the main goal of this carbohydrate metabolism. So yeah, that is it. I think this presentation makes sense, and okay, thank you.